Alright, so in this video we're going to go ahead and talk about cells. In particular, we're going to talk about the different types of cells. We're going to talk about how they utilize and how they're made out of biomolecules, and then how we actually transport those molecules. So first off, the different types of cells that we see. The first one that we're looking at in this picture down here at the bottom is a prokaryotic cell. Remember, pro, the prefix, actually means before. Karyotic means nucleus. So a prokaryotic cell is before a nucleus. That means that it is obviously extremely old. It's the first type of cell that we see, and it is extremely simple. It doesn't have a nucleus. That means it also does not have organelles. So let's look at what it does have. It has DNA, and we know that DNA is super important because DNA is basically the code for making proteins. We know that it has ribosomes. Oh, sorry, DNA, point to it. Ribosomes, those are the tiny little dots in there, and those are the actual building factories for proteins. So DNA codes for proteins, and then the ribosomes actually go out and make them. It has a plasma membrane. The plasma membrane is basically, imagine like there's a Ziploc baggie around everything that needs to go inside. So the plasma membrane holds it all together, and it allows certain things to enter and leave. And lastly, it has cytoplasm. Cytoplasm is this jelly stuff in the center of the cell. Imagine that, okay, so if the plasma membrane were like a Ziploc bag, the cytoplasm were jello filling that bag. Cytoplasm is extremely important because it prevents the internal stuff from sloshing around too much. Alrighty, so there are really only 4 things that fill a prokaryotic cell that you need to remember. What's an example of a prokaryotic cell? That would be bacteria. Alrighty, up at the top, we've got 2 cells. Each one of them is crazy looking on the inside. They have so much stuff going on. Both of these are eukaryotic cells. The prefix of eu means true. So it means true nucleus. So let's go ahead and look at what stuff fills the inside of the cell. The first thing is plasma membrane. And notice, plasma membrane is also in bacteria. So again, plasma membrane, that's the baggie that holds everything together, and it'll also control what enters and what leaves the cell. The next thing they both have is cytoplasm. Again, jelly stuff filling the inside. Again, bacteria also has that. DNA. Bacteria also has that. Ribosomes. Again, little factories that make protein. Eukaryotic and prokaryotic both have these. There's one difference though. Some of the ribosomes in eukaryotic are free-floating, meaning they're just floating in the cytoplasm like that. Or some of them can be bound on organelles. So right here, what I'm circling in the plant cell, those ribosomes are all stuck to an organelle. So as of right now, you can see that there are 4 things that I have in red boxes. Those are the 4 things that pro and eu all share in common. How are you going to remember this? I always say, don't hate runny chocolate chips. So the D stands for DNA. The H stands for homeostasis. The word homeostasis means balance. So all living things have the ability to maintain homeostasis, meaning they move stuff in, they move stuff out of their cell to keep themselves balanced. The R stands for ribosomes. The C stands for cytoplasm, and the last C stands for cell membrane. Alrighty, so those are the 5 things they have in common. Let's go ahead and look at what eukaryotic cells have different. They have organelles. So remember, pro is super old and simple. They don't have organelles. Eu, on the other hand, is newer. It's complex. They have organelles. So again, eu, complex. Alrighty, so the first organelle I have on the list is the nucleus. We know obviously that the nucleus contains the DNA inside of eukaryotic cells. That's one of the major things that makes it different. The next one is endoplasmic reticulum. The endoplasmic reticulum, I'm going to go ahead and circle in yellow. It's this structure right here, so the one I'm circling in yellow. So the endoplasmic reticulum, its job is it can either be covered in ribosomes or it can be without ribosomes. So it can either be smooth or rough. its job is to produce proteins, package proteins, and then lipids. So because it has ribosomes, and I know that ribosomes build proteins, it is part of that process of building and making and packaging proteins. All right, the next one is the mitochondria. I'm going to go ahead and circle this in red. Mitochondria are extremely important because they actually take food and break it down. They do the process of cellular respiration to make energy for us. The next one is the Golgi apparatus. The Golgi apparatus, which I'm circling in purple, it is important because it takes all of the proteins, all of the lipids that my endoplasmic reticulum made, and it will actually package them, it'll edit them, and it'll send them to different places in either the cell or to different cells. The next one is the chloroplast. Chloroplast is only going to be found in plant cells. It looks like it has a tiny little stack of pancakes inside of it. And its job is to do photosynthesis, meaning it takes solar energy, it stores it in the bonds of glucose in food. And the last, let me erase this. The last two that we're going to look at is going to be the vacuole. The vacuole has a specific job in plants where it is for storage. So it's that big blue, it's the big one that I outlined in blue. Its job is basically to hold on to extra food, extra water, in certain cases waste even. All right, and the last thing is the cell wall. So cell walls, they're only in plant cells. Animal cells don't have them. One thing we'll talk about in a second is that the cell wall is actually made out of biomolecules. We'll talk about that in a sec. Prokaryotic cells also have a cell wall. So that is not something exclusive to you, cell walls. Plants have them, bacteria have them, animals do not. So how did I get these extremely complex cells? The theory of endosymbiosis tells us that a small prokaryotic plus a large prokaryotic, so again, this one is pro, this one is pro, they combined. When they combined, the small one was engulfed, meaning it was swallowed by the large one, and instead of dying and getting broken up like food, it survived and it lived on the inside of the bigger prokaryotic cell. Well, after hundreds and thousands of years of that kind of relationship continuing, we ended up getting a totally new kind of cell called a eukaryotic. So the theory of endosymbiosis says pro plus pro gave me you. We say today, that organelles like the mitochondria, like the chloroplast, used to be tiny prokaryotic cells. Why? Because they have their own DNA. So if I look inside of a chloroplast or mitochondria, it'll have DNA. Tiny little bacteria used to have its own DNA too. We say that chloroplast and mitochondria can reproduce on their own. So if I need more of them inside my cell, it will copy itself by itself without the cell copying it. And then we also say that it's about the same size as bacteria is today. So if I were to compare this tiny bacteria to the size of my mitochondria, it would be about the same. Alrighty, so the next thing we need to talk about is what biomolecules make up my cell. The first one is nucleic acids. Obviously, we know that DNA is super important in our cells because it programs and it tells my cell how to make proteins. Without protein, we know that our cell would basically not function and we wouldn't work and our cells would die. The next one is carbohydrates. Carbohydrates play a couple of important roles. The first role is that they are made in the chloroplast and they actually store energy. So we always talk about carbohydrates being quick energy. The other thing that they're really important for is they actually make up the cell wall. So the carbohydrate cellulose is stacked all together and it again makes up the cell wall in plants. The next one is proteins. Proteins have a couple of different jobs. One of the jobs, for example, is they are enzymes. Almost misspelled. Alrighty, so for enzymes, for example, RNA polymerase, they play an extremely important role in different processes such as making RNA. We know making RNA is the first step in building a protein. So if I didn't have this protein, RNA polymerase, I wouldn't be able to make RNA and I would not be able to then read the RNA and build a protein. So if I don't have proteins like enzymes, I can't make other proteins. So they play an extremely important role in the cell. And then we also, when we look at proteins, we're also talking about how they play an important role in transport. And we see that when we look at lipids. So lipids make up the cell membrane. So right here, oops, sorry, that was white. This right here, that is part of the cell membrane. That's made out of lipids. But these guys right here in the cell membrane, those are proteins. So going back really quick for proteins, they're very important in transport. They're very important, transport to molecules. They're also very important in making other proteins. They're important to be enzymes, to break stuff, to make stuff. They're just important all the way around. Proteins also give us like hormones, for example. They're also going to be muscle. So they've got a lot of really important jobs. We usually just say they do work. Alrighty, for lipids now, I said that one example of a lipid is your cell membrane. So you do need to know that your cell membrane is made out of lipids. Lipids also store long-term energy. They give us insulation, they're fat, they're blubber, they keep us warm. There are a bunch of different jobs that lipids do. The last thing we're going to talk about is how do I move molecules. So this line in the middle, again, that is the cell membrane, which is made out of lipids. And notice how the cell membrane moves stuff in and out. Why do I want to do that? Because I want to maintain something called equilibrium. Equilibrium means balance. There's another word we use for that word, and that word is homeostasis. So you need to know both of these words, equilibrium and homeostasis. They both mean balance. Okay, so basically my cell doesn't want a lot of waste to build up. It also doesn't want a lot of food to be missing. Like, I need a balanced amount of everything inside of me. Alrighty, so when we talk about the cell membrane is two things. The first thing is semi-permeable, and the second thing is selectively permeable. The word semi-permeable means that there are certain things that can go in and out. Physically, things can fit through the membrane while other things cannot. The second word, selectively permeable, means that the membrane actually selects what it wants to allow across the membrane, meaning there will be certain times where it doesn't let stuff into the membrane. There will be other times where it does let stuff across the membrane. It is selective. Okay? So let's talk about the different types of transport that I have. Remember, we always draw the hill. We say low, high, low. If I'm going up the hill, I need ATP. Imagine you are climbing a hill. You will need energy. If I'm biking up a hill or something like that. We call moving up the hill from low to high active transport. So again, low, high, moving across the membrane, and I'm using ATP. Notice active, ATP, they all start with the letter A. If I'm moving down the hill from high to low, on the other hand, I do not require ATP. No ATP. So on this side, we see low, we see high. I'm moving across from high to low. This is called passive transport or diffusion. They both mean the exact same thing. Notice in the second picture, though, I'm still going high to low, but now I'm going through a protein. That protein, though, is not using ATP like it is in active. So I'm going high to low. I'm not using ATP. It's still diffusion. It's still passive transport, but we call that facilitated, meaning I need a protein to help me. Why would I need facilitated diffusion if I'm moving something that's kind of chunky and large, but it's still going from high to low? I would also need a facilitation protein when I'm moving things in a kind of a quick manner and I need to get them across the membrane and they're larger or they're charged. You'll talk about charged particles in chemistry. I'm not going to worry about that too much with you guys. There's one more thing you need to know. Whenever we talk about the hill, if we say that we're going up the hill, we're doing active transport, we could say the words up, or we could say the word against the gradient. Keep that in mind. If I'm going down the hill, I could obviously use the word down, or I could say with the gradient. So keep that in mind. That's just two different ways that they could describe moving up or down the hill. Alrighty, the next thing we're going to talk about for active transport, just a quick second, is what if I want to move a bunch of stuff in or out all at the exact same time? Well, if I want to move a bunch of stuff out, I form a vesicle. So a vesicle. and it is basically just plasma membrane surrounding whatever I want to take in and out of my cell. That plasma membrane is going to join with the vesicle, and basically it'll open up the vesicle and let all of the stuff that I was carrying out. This requires ATP because I'm moving a bunch of stuff out of my cell all at once. Exo means exit. Endocytosis, on the other hand, means I want to bring a lot of stuff in. So my plasma membrane begins to form a little bubble around everything I want to bring in. Eventually it pinches off and it forms a vesicle that will float and go inside of my cell with all of the stuff I want to carry. This, again, bringing a lot of stuff in all at once, requires ATP. So these are both a form of active transport. They are just examples of when I want to bring a lot of stuff or very large stuff all in at once. All right, last thing we need to talk about is osmosis. Osmosis is the movement of water. It is a type of passive transport. So for water, if I look in this picture right here, in picture A, and then I've got picture B. In picture A, I have a lot of free-floating water, meaning it is not bound to anything. And let's say, for example, there's actually 24 molecules of water. I don't have anything else in the picture. In picture B, I have these little salt molecules, or sugar, whatever it could be. I call that a solute. Salt, sugar, those are solutes. Notice here I only have 1, 2, 3, 4 molecules of free water. Water likes to go where there is less free water. Because in picture A, there's so much free water, it's bumping into itself. It doesn't like that. It wants to go where there's less free water. So water will move from high water to low water. The easier way to think about this, though, instead of thinking about it in terms of water, is actually to think about it in terms of salt. So basically, water will follow the solute. I like to say salts. Okay? So in other words, water will go to where there's more salt. If all the water starts to fill up a cell, that cell will start to grow in size just like a water balloon. If it eventually keeps growing, it will pop. If all of the water leaves a cell, all of the cell will begin to shrivel up and it'll kind of look like an uninflated water balloon. If water goes in and out equally, then that's what I want. That's the goal. My cell will stay the same. There are 3 types of solutions in which water can move. The first one is hypertonic. Hyper means above. So if Miss Anda's really hyper, she's acting above what she should be acting. That means there is a high amount of salt outside. That means there is a low amount of salt inside the cells, and water follows the salt. So water will leave the cells and my cells will shrivel. This will also happen in plants. Notice the cell wall in plants stays the same size. Alrighty, so the cell wall stays the same size, but the plasma membrane, the cell membrane on the inside, does shrink. So plants can have this happen to them as well. The next one is isotonic. Iso means equal. That means there is, let's say, there's 75% salt outside, there's 75% salt inside, water moves in, water moves out equally. This is perfect. This is what I want. The last example is hypotonic. For hypo means below. So that means, let's say that it is low salt outside, it is high salt inside the cells, and so therefore my water will move in. Again, water follows the salt. If all the water fills into the cells, it's just like a water balloon that I keep filling until it bursts.